And hi, everybody. I'm just going to quickly share my screen. And this is the part where things usually go wrong. So everybody cross your fingers. That is going okay so far.
Yep.
There we go. You can see all this, can't you?
Yep. Yes.
Lovely. And hopefully my ugly mug is no longer on the screen. So today I'm going to be discussing what we mean when we talk about pathophysiological mechanisms and shifting them across to therapeutics. So my name's Simon. I'm the Director of Research at Cure Parkinson's. And when we talk about pathophysiology, we are talking about a branch of science that is the intersection between pathology and physiology. Physiology being normal behavior for organs or biological structures, and pathology being the opposite, basically. And when things go wrong, it's a really tough process of actually working out the mechanisms of what's actually gone wrong. Is it at the whole organism level, or is it down at the cellular or microcellular level? But with regards to Parkinson's, I thought what I'd do is start off by providing a bit of context, a bit of history with regards to how we got to where we currently are, and that'll hopefully put everything into context for you and provide you with a good background as to some of the discussions that are going to be had over the next 2 days. So this graph here, this is a slide I use quite often. It shows you all of the research that's ever been done on Parkinson's in terms of research papers published each year. And in 2022, there were 11,608 scientific reports published. And in total, at the end of 2022, we had 161 research reports published, and almost 80% of that's been published since the year 2000. But today, we're not going to start with James Parkinson's description of the condition, because that was clinical. That's what the disease looked like when a person presents at a doctor's office. Rather, we're going to go down into the biology, and the history of the biology really starts here in the year 1912. And the chap of interest in this picture is the one with the red circle around his face. This is Frederick Louis. I'll explain why he's important in a moment, but I'll also just point out that this is a photo of some pretty eminent people, including this chap here, who is Elias Alzheimer's. And this is actually an interesting picture because it's one of the only photos of Elias without a cigar in his hand. He was a man for cigars. The chap beside him is holding a cigar, but Elias in this particular photo is not. It's one of the only photos where he's not holding a cigar. Anyways, useless pub quiz detail there. Frederick Louis, at the time, was examining post-mortem sections of brain from people who had passed away with Parkinson's. So they were quite advanced with their Parkinson's. And what he noticed were these tiny little spherical inclusions inside neurons. And these ultimately adopted his name. You can see here in the photo with the yellow arrows pointing, there's a sort of a circular, fuzzy kind of circular body. It's separate from the nucleus, which is behind the yellow arrow. But these are quite a characteristic hallmark of the brains of people with Parkinson's. Not everybody has them. In fact, some people without Parkinson's have Lewy bodies as well. But at the time, the research world got very excited because this was one of the first biological structures associated with this particular condition. And then a few years later, Konstantin Tretyakov, he noticed in the substantia nigra region of the brain, which is kind of at the top of the brainstem, but beneath everything else, that there was an absence of neurons in this area called the substantia nigra. And ultimately, we'll go on to see that the substantia nigra is quite important, but this was the first time anybody noticed that the neurons in this particular region were missing or absent in people with Parkinson's. And they're important because these are the dopamine neurons, but at the time they didn't know what dopamine was. That came in 1958 with this chap here, Swedish scientist Arvid Carlsson. He discovered that the chemical dopamine is a neurotransmitter. So it's a protein that communicates from one neuron to another. And he actually went on to win the Nobel Prize in medicine a few years back for this. But it was this guy here that really sort of brought all the pieces of the puzzle together. This is Ole Hornikowitz. And he noticed that in people with Parkinson's, this chemical dopamine was significantly depleted. You can see on the left-hand side of the screen here, the original figure from one of the original figures from the paper he published. And there are two structures that he analyzed called the caudate and the putamen. And in the caudate, there was in people with Parkinson's who passed away with Parkinson's, he found only 20% of normal levels of dopamine. And in the putamen, there was only 2%. There are some, oh, who's joining the room here? I've got somebody joining the room virtually. But the following year, Uli and his collaborator, Walter Birkmeyer, they decided to start injecting people with levodopa, people with Parkinson's. If there is an absence of dopamine in people with Parkinson's, why don't we inject them with levodopa, which is one of the critical ingredients in the production of dopamine, and we'll see what happens. And what happened can only be described as miraculous. They wrote in their journal that bedridden patients who were unable to sit up, and patients who could not stand up when seated, and patients who, when standing, couldn't start to walk, all performed these activities with ease after L-Dopa. But the L-Dopa treatment that these chaps were using was an injected L-Dopa, and it was just straight L-Dopa. There was nothing else, and so there were side effects associated with this. The body was breaking down the L-Dopa before it could get to the brain, and it wasn't until 1968 when George Kotzier really sort of perfected the use of oral levodopa in combination with carbidopa, that they were able to get enough L-Dopa to the brain of people with Parkinson's that became a viable treatment. Now, none of this is going to be on the exam, but I just wanted to give you a sort of a background so you can sort of see the history stretching out over the last 50 years. And in the 70s, we had the approval of levocarbidopa. And then we also had in the 1980s, we had the first dopamine agonists approved by the US FDA, followed by MAO-B inhibitors in the mid-90s. And in 1997, the FDA approved deep brain stimulation. But the important thing to remember about all of these treatments, in fact, all of these treatments here, I've stolen this slide from Michael J. Fox Foundation. They're nice people, so they'll forgive me. But it gives you, you can see the spread and the development of these treatments over time, back from the 1950s and 60s all the way through to the modern era. And most of them, the dark blue bars, most of these are motor symptom specific treatments. But gradually, as you get into the 2000s, you start to see orange bars, and this is where the non-motor treatments are coming through more and more and more. But it's important to remember all of these medications are symptomatic medications. So these are medications that cover up the symptoms of the disease, but they don't actually do anything with regards to the cause or the driving mechanism of pathology. And that's where the last few years have been quite interesting for us, the more recent history. And it starts in 1997. So when most of the world were excited about Leo and Kate and Titanic, or Dolly the sheep, or a young man named Tiger who was tearing up the golfing world, and a radio-controlled car on Mars, The Parkinson's research world were really, really excited by the research of this man. So this is Lawrence Colby, Professor Lawrence Colby, and he found a family in Italy where 61 members of the extended family of 574 had developed Parkinson's. And this is a extremely frequent, high frequency of Parkinson's within a specific population of people. So he collaborated with a group of researchers, and they determined that there was a genetic variation, so a tiny error in the DNA of all the affected family members, and it lay in a region of the DNA that we call the alpha-synuclein gene. So this is a region of DNA that produces a protein, that provides the instructions for making a protein called alpha-synuclein. And lo and behold, 3 months later, researchers in Cambridge University here in the UK, collaborating with a group in Philadelphia, they found that alpha-synuclein protein is actually sitting in those small spherical bodies that Frederick Lurie first identified way back in 1912. So you have Lurie bodies, which are the hallmark of people with the brains of people with Parkinson's, and you connect alpha-synuclein to those Lurie bodies, suddenly you're starting to see potential mechanisms of pathology creeping into the overall picture. So these everybody got very excited in the early 2000s as a result of this connection, and what can only be described as a gold rush of genetic sequencing ensued. And over the next few years, they discovered more genetic risk factors associated with Parkinson's. I should say here that Parkinson's is not a genetic condition. These risk factors only impact about 1 in 10, or approximately 1 in 10 cases of Parkinson's. But we've found about 80 of these genetic risk factors. And what's really interesting is that it points towards associated biology. So each of these risk factors is in a particular region of DNA that's involved with providing the instructions for a particular protein. And when researchers look at the biology associated with that protein, they start to get an idea of what could be impacted in people with Parkinson's. And so the early 2000s was a very busy period of establishing that biology that was associated with these genetic risk factors. And then in the 2010s, we had a better understanding of that biology, and it was pointing towards pathophysiology that could be associated with Parkinson's. For example, one genetic variation occurs, it's a very common genetic variation for people with Parkinson's. About 1 in 10 people with Parkinson's, in particular populations of people, can carry a gene, genetic variant called in the GBA1 gene. And this is associated with a protein that's involved with waste disposal in cells. And people that have this genetic variation have a reduction in the levels of that enzyme. So suddenly researchers are being presented with the possibility of actually modulating some of this biology that's associated with these genetic risk factors, and asking themselves, could this potentially have therapeutic effects if we correct the biology that's affected? Could we be slowing down the progression of Parkinson's and improving quality of life for people with the condition? So the 2010s was a period of drugs discovery, drug screens, and drug development based on some of these biological pathways. And now in the 2020s, it's a very exciting time for the research community, because we have biotech firms and pharmaceutical companies that are lining up clinical trials that are targeting these biological pathways with particular agents. And hopefully some of these more targeted experimental drugs will provide better treatments going forwards for people with Parkinson's, ideally slowing down the progression of the condition. And I just wanted to give you an example of one of these. There's a protein called leucine-rich repeat kinase 2, or LRRK2, and it has lots of functions in the cells of our bodies. It's a very busy protein. It does lots of things. But approximately 1% of the population of the Parkinson's population carry a genetic variation in their LRRK2 gene. So the region of DNA that provides the instructions for making LRRK2 protein. And the result of that genetic variation. Oh, Gary's joining us. Hi, Gary. No, he hasn't joined us yet. Somebody needs to admit him into the room. Anyway, LRRK2, the genetic variation associated with LRRK2 results in a very hyperactive form of the protein. Think bull in a china shop sort of scenario. And so researchers have been asking, if we tone down, if we inhibit that hyperactivity of LRRK2, could that slow down the progression of Parkinson's? And one of the biotech companies that's been leading the way here is a company called Denali Therapeutics. And in 2021, the company announced the results of phase 1 testing of their molecule DNL151. They tested it in 184 healthy volunteers and 36 people with Parkinson's. This is a first-in-class drug. It's the first time anyone's ever tested a LRRK2 inhibitor in humans. And so they've been very, very careful in how they progress. So they've tested a lot of people, healthy volunteers, and also 36 people with Parkinson's over a month. And they found it to be, the molecule to be safe and generally well tolerated. But more importantly, they found that it actually did what it said on the label. It inhibited LRRK2. If you look at the graph on the left-hand side of the screen here, the gray line going sort of horizontally across the middle of the picture is the placebo-treated individuals. So the LRRK2 activity in these people did not change very much. It just sort of stayed the same across the 28 days of treatment. But then there's a yellow line and a red line and a blue line, and these are different doses of the LRRK2 inhibitor. And what you notice, hopefully, is that these lines all go sort of crashing down towards the bottom, resulting in more than 75% inhibition of LRRK2 activity in most cases. And it's stable across time as well, across the entire month of treatment. Excuse me. And interestingly, you also see the same reduction in activity when you look at the proteins, some of the proteins that LRRK2 interacts with in cells. So one of those proteins is called RAB10. And when you look at RAB10 activity, you can see the same thing as on the right-hand graph. You see the same thing as you see in the left-hand graph. The gray line across the middle, again, is placebo-treated individuals. And then the yellow line, the blue line, and the red line are all crashing towards the bottom of the graph, suggesting that RAB10 activity is reduced as well. So this is very exciting. It suggests that the drug is safe, firstly, and that it's doing what it's supposed to do. And so this got another biotech company called Biogen to partner up with Denali, and they started a phase 2B clinical trial last year called the LUMA study. And this study is currently recruiting, they're looking for 640 participants with early Parkinson's who will be treated blindly with either placebo or the LRRK2 inhibitor, which is now called BIIB122. And they'll be treated for 48 weeks, potentially 2.5 years if things go well, but 48 weeks will be sort of the primary readout of whether it's having an effect, or the initial readout of if it's having an effect. So this is just an example of where we've gone from pathophysiological mechanism, that being the hyperactive behavior of a particular protein in people with Parkinson's, some people with Parkinson's, and identified a molecule that can modulate that pathophysiological behavior. And hopefully that will lead to a therapeutic intervention in the not too distant future. And one question you might be asking yourselves is LRRK2 activity increased in people with idiopathic Parkinson's? So it's a person who does not have a genetic variation in their LRRK2 gene. Could a LRRK2 inhibitor be useful in these individuals? And the answer is potentially yes. Professor Tim Grenemeyer at Pennsylvania University and collaborators had a look at people with idiopathic Parkinson's, and they found that LRRK2 activity was elevated in postmortem sections of brain from these individuals in vulnerable regions of the brain. And not only that, but also, again, this protein that LRRK2 interacts with, RAB10, that was also elevated levels of activity. So that suggests that people with idiopathic Parkinson's might also have elevated levels of LRRK2 activity, and LRRK2 inhibitors might be useful for a much wider portion of the Parkinson's community than simply those with a genetic variation. And this has resulted in a lot of interest from the pharmaceutical world. The biotech companies, in this picture here, I've got a few of the biotech companies that are currently developing LRRK2 inhibitors or LRRK2 inhibitor programs for Parkinson's. And Neuron 23 has gone to trial, and the OPM OncoDesign has also gone to trial. And I believe GlaxoSmithKline has also got a clinical trial program active as well. So there's quite a lot of activity as a result. So it's quite an exciting time. But just I wanted to give you an example of how we're moving from pathophysiological, an understanding of the pathophysiological mechanisms through to therapeutic interventions. So to summarize, as we said at the start, pathophysiology is the intersection between pathology and physiology, where things are normal and how things go wrong. And genetic risk factors have pointed towards associated biology that could be implicated in the development and progression of Parkinson's. Researchers have now identified molecules that can modulate or manipulate that biology, and a number of clinical trials are now up and running, and we'll hopefully be hearing a bit more about that over the next 2 days. And this is how we move from pathophysiological mechanisms to therapeutics. And that's all I have to say today. I'm going to say thank you very much. And